Hello everyone. Welcome to today's webinar on the Illumina DNA Prep Tagmentation Library Preparation Kit. This webinar is meant to be an introduction to this kit, so this presentation will be geared more towards customers who are just getting started rather than those who have been running this library preparation workflow on a regular basis. Before we start, I would first like to discuss a name change recently associated with this product. Illumina DNA Prep Tagmentation is the new name for the Nextera DNA Flex kit. So if you've previously used the Nextera DNA Flex kit, this webinar is about the same product but with a new name. The M refers to the medium insert size compared to other kits, and tagmentation refers to the method of input fragmentation. Please note that some of our published documents, such as application notes and data sheets, still use the Nextera DNA Flex name, so you may still see this name in some figures throughout the protocol. Again, Illumina DNA Prep Tagmentation and Nextera DNA Flex both refer to the exact same product. Some associated index kits have also been renamed, and we will discuss this later in the webinar. For the library prep kit, the protocol has not changed. The catalog numbers and reagent composition have not changed, only the name has changed. I also wanted to make a note that the name of this kit is very similar to the Illumina DNA Prep S Tagmentation Kit, which is part of our Illumina DNA Prep with Enrichment Kit. This is a separate enrichment workflow that will not be discussed in this webinar. Now let's get started with the presentation. First, what is Illumina DNA Prep Tagmentation? Illumina DNA Prep is a library preparation kit that offers flexibility for many whole genome sequencing applications. One popular application is for human whole genome sequencing. However, the kit is also compatible with genomic DNA from non-human species, including large or complex animal or plant genomes. This opens up applications such as agrigenomics or sequencing with your favorite model organisms like Drosophila and mice. Additionally, the kit is compatible with small genomes, so it's useful for metagenomics or public health research and surveillance applications. Illumina DNA Prep also has the flexibility to accommodate variations in sample type, DNA input amount, and applications. Later on in this presentation, we will discuss alternative types of input that can be used beyond genomic DNA. Illumina DNA Prep is one of the fastest Illumina library preparation workflows with a total prep time of under 4 hours. This kit has optimized library prep performance, generating reliable results. Here is the outline of today's webinar. First, we will discuss the advantages of using the Illumina DNA Prep kit. We will then talk about the consumables and equipment needed for library preparation. We will go into input recommendations, the library prep workflow itself, library quality control, and sequencing considerations. And then we will also delve into the optional blood, saliva, and bacterial workflows. First, let's discuss the advantages of using Illumina DNA Prep. Illumina DNA Prep has improved coverage uniformity as compared to previous tagmentation-based kits. This kit utilizes enzymatic fragmentation instead of mechanical fragmentation, which is used in our TruSeq kits. This is useful and very convenient for labs that don't have access to a covaris for mechanical shearing. In this graph, we see a comparison of human genome coverage with Illumina DNA Prep, which uses enzymatic fragmentation, and TruSeq DNA Nano, which uses mechanical fragmentation with covaris. As you can see, coverage between these two kits is very comparable. Illumina DNA Prep has been optimized to work well with low and high GC content genomes. On the left is an organism that has low GC content, and on the right is an organism that has high GC content. You can see improved coverage over previous versions of tagmentation-based kits, such as Nextera XT. This is especially helpful for metagenomic sequencing applications, where we may be dealing with a range of microorganisms with variations in GC content. These figures can be found in our Illumina DNA Prep datasheet, and you can find the link on this slide. Illumina DNA Prep is also compatible with a wide range of DNA input quantities. If you're familiar with our Nextera XT kit, you may know that the input requirement is exactly 1 nanogram. For Illumina DNA Prep, we have more flexibility in the input. Here in this image, you can see that we have validated input ranging from 1 to 500 nanograms. This graph shows insert sizes with varying amounts of DNA input on the x-axis. The insert size on the y-axis is very comparable between 1 nanogram input and 500 nanogram input. Another nice feature of this kit is that if we start with between 100 to 500 nanograms of DNA input, library yield is normalized. Here on the y-axis is the yield in nanograms per microliter, and you can see that the yields are comparable when we use an input range between 100 to 500 nanograms. This normalization of library yield is also expected if we use the Lysis protocols for liquid blood or saliva, or the demonstrated protocols for dried blood spots or microbial colony extraction. We expect these alternate protocols to generate enough starting input DNA to allow for library normalization. We will be discussing these alternate protocols later on in the presentation. In this slide, I will be comparing Illumina DNA Prep with our older Nextera XT kit, which is still available. In terms of the type of input, Nextera XT is only compatible with small genomes, while Illumina DNA Prep is compatible with both small and large genomes. As we discussed in a previous slide, Illumina DNA Prep is compatible with 1 to 500 nanograms of DNA input, although for large genomes, we do recommend at least 100 nanograms of input. With Nextera XT, we require exactly 1 nanogram, no more and no less, so Illumina DNA Prep is more flexible. Another difference is in the tagmentation reaction. With our tagmentation-based kits, double-stranded DNA is enzymatically fragmented with a transposome. With Illumina DNA Prep, the tagmentation reaction occurs on beads, while with Nextera XT, the enzymatic reaction occurs in solution. The next difference is with PCR. With Illumina DNA Prep, since we start with a variable amount of DNA, we will have variable cycle numbers based on the input quantity. With Nextera XT, we have a fixed number of cycles since we always start with 1 nanogram input. The final difference in the protocol is with the post-amplification bead cleanup step. Illumina DNA Prep uses a double-sided side selection, which results in a more narrow library size distribution. Nextera XT, on the other hand, uses one-sided side selection and consequently has a broader library size distribution. The narrower size distribution of Illumina DNA Prep makes it easier to optimize loading concentration for sequencing. Next, let's talk about the consumables and equipment that are needed before starting library preparation. There are 3 categories of products needed for Illumina DNA Prep. The first is the optional Flex Lysis reagent kit. This kit is only required if you plan on using blood samples directly with the kit. It cannot be used with other types of input, only with blood. Next, we have the Illumina DNA Prep M Tagmentation Library Prep reagents, which are required for this workflow. These reagents come in either a 24 sample or 96 sample format. Please note that the required index kits are purchased separately from the library preparation reagents to allow for more choices in indexing. There are several indexing options. The first are the combinatorial dual or CD indexes, which come in a 24 sample or 96 sample format. We also have the option of using unique dual indexes or UD indexes with the Illumina DNA Prep kit. So what is the difference between combinatorial dual and unique dual index kits? With CD indexes, index combinations can share either an I5 or an I7 sequence, even though the index combination is distinct for each sample. With UD indexes, index combinations have unrelated index sequences for I5 and I7 index reads. To illustrate, with combinatorial indexes on the left, when we go across the plate, we will have repeated I7 indexes across each row and repeated I5 indexes down each column. However, when we look at the final library index colors below the plate, we can see that even though some indexes are repeated, green in this case, each combination of indexes on the plate is unique. On the right is an illustration of unique dual indexing. In this setup, there are no redundancies in the I5 or the I7 sequences in the entire plate. Each I5 is unique from all other indexes, and each I7 is unique from all other indexes. Unique dual indexing is especially useful in mitigating possible index misassignment from index hopping. With UD indexes, any index hopped reads will be bioinformatically removed. Here are the 4 unique dual index kits available for Illumina DNA Prep. Each set has 96 single-use wells with premixed I5 and I7 indexes. All 4 index sets could be combined on a run for up to 384 uniquely indexed samples. Here, I do want to note a name change for these indexes. Sets A and B have changed from Nextera DNA Unique Dual Indexes to IDT for Illumina DNA RNA UD indexes. The catalog numbers and the indexes remain the same. The names of sets C and D have not yet changed because later this year we are planning on releasing version 2 of these kits. For version 2, the sequences of several indexes will change. All these indexes can be found on the Illumina DNA Prep product page, and the index sequences can be found in our adapter sequences document. Now that we have our reagents, let's ensure that we have all the equipment and consumables that we need. In terms of equipment, we would need a microcentrifuge, MIDI plates, a magnetic stand that is compatible with 96 well MIDI plates, a vortexer, and a thermocycler. If you're using the blood or saliva sample workflow, these are potential biohazards. For these workflows, we will need a water bath and an additional thermocycler, which should be located in a BSL-2 lab or cabinet. For more specifics on consumables and equipment needed, including recommended thermocycler models, consumable suppliers, and part numbers, you can find these on pages 27 to 29 of the linked reference guide. While we're on the topic of reagents, I would like to mention one more thing before we move on. Illumina DNA Prep is typically used for whole genome sequencing and has not been validated for the ATAC-se method. ATAC-se is a popular method for profiling accessible chromatin, and it uses tagmentation reagents from the discontinued Nextera DNA kit. If you're working with a Nextera DNA tagmentation enzyme and buffer for a custom application that uses free transposomes, such as with ATAC-se, please do not substitute Illumina DNA Prep reagents, as they have not been tested for these applications. Instead, please use the Tagment DNA TDE1 enzyme and buffer kit. The catalog numbers for the large and small kit sizes are shown below. Now that we know how to get the correct equipment and consumables, let's go over input recommendations for Illumina DNA Prep libraries. As we discussed earlier, Illumina DNA Prep supports a variety of sample input types. Apart from genomic DNA, this protocol is also compatible with cDNA samples, plasmids, or PCR amplicons greater than 150 base pairs in size. Input DNA needs to be double-stranded to be compatible with the tagmentation process. We can also use direct input from blood and saliva using the supplemental procedures at the end of the protocol. We also have demonstrated protocols available to use input directly from microbial colonies or from dried blood spots, and these protocols are linked here. If we are working with a small genome, including most bacteria or even plasmids and PCR amplicons, we can use anywhere between 1 to 500 nanograms of input. If we're working with large genomes, including eukaryotic genomes or genomes greater than 5 megabases, we recommend at least 100 nanograms of input. This larger input amount is needed to capture the complexity of larger genomes. Even though Illumina DNA Prep provides a large range of flexibility of input sample types and DNA input quantities, it's important to note that users still need to know generally how much DNA we're putting into the assay. If we have an input that is known to be between 1 to 100 nanograms, quantification with a fluorometric assay such as qubit or picogreen is required in order to determine the number of PCR cycles. If we know that our input is somewhere between 100 to 500 nanograms, accurate quantification is not strictly required. An advantage of Illumina DNA Prep from previous tagmentation-based assays is that it is less sensitive to the accuracy of input quantification at 100 to 500 nanograms. Looking at the table below, we can see that the number of PCR cycles required is variable between 1 to 100 nanograms of input. However, once we reach 100 nanograms, we will always use 5 PCR cycles. I would also like to emphasize that if we start with at least 100 nanograms of input, we expect normalized library yield. In order to have normalized library yield from fresh blood or saliva, we will need to use the fully integrated blood and saliva lysis protocols in the supplemental procedures. We also recommend qualifying the input using a UV-based method like nanodrop to determine if the 260 over 280 and 260 over 230 ratios are within our recommended ranges. Ratios outside these ranges may indicate contamination, and contaminants in the input may inhibit library preparation. On this slide, I've provided a link to a bulletin describing these possible contaminants. Additionally, Illumina DNA Prep is optimized for high-quality input, and use of fragmented or degraded DNA may lower overall library yield. If you're planning on using degraded input, reach out to us in tech support. Now that we have the correct type of DNA input and the correct quantity, we can start library preparation. Here is an overview of the library preparation workflow, and we will discuss this in more detail in the next few slides. We will start the protocol either with genomic DNA or alternate inputs such as cDNA, plasmids, or amplicons. Or we can start with the optional direct blood lysis or saliva lysis workflow. We will talk more about the blood and saliva lysis protocols later, but once we have lysed the sample or diluted our DNA, the workflow will be the same from this point on. The DNA from this first step will be enzymatically fragmented or tagmented into smaller pieces. After tagmentation, the fragments will be cleaned up. This will be followed by an amplification step, which increases library yield and also adds index adapters by PCR. After this, we will clean up the libraries to narrow the size distribution. And finally, we will pool the libraries for sequencing. On this workflow, validated safe stopping points are indicated. If there is no safe stopping point at a certain step, we recommend continuing with the workflow immediately. These safe stopping points are something to keep in mind when you're planning the timing of your experiment. Now, let's talk about the first step in greater depth, Tagment Genomic DNA. When we talk about tagmentation, we mean that the DNA will be both enzymatically fragmented and will also be tagged in a single step. As we discussed earlier, something new about Illumina DNA Prep is that tagmentation occurs with bead-linked transposomes, or BLT, rather than with free transposomes. Here, we see that the transposome enzymes in yellow are attached to the beads. The genomic DNA will be captured by the transposomes, and it will be enzymatically fragmented and also tagged with the partial adapter sequences represented in blue and green. This step happens on a thermocycler for 15 minutes at 55 degrees Celsius, so it's a very quick step. Here, we can also see why higher input is normalized with bead-linked transposomes. At a certain input quantity, the beads become saturated with DNA, and any additional DNA will be washed away. Now, let's talk about a few best practices for handling bead-linked transposomes. The bead-linked transposomes are magnetic beads, and these have to be stored between 2 to 4 degrees Celsius. We need to ensure that the beads are not frozen. If we do accidentally freeze the beads, the efficacy will be greatly reduced. We also want to ensure that we vortex the beads to fully resuspend them before use. Centrifugation is not recommended before pipetting. However, once the beads are in the sample, we can briefly centrifuge the plate if the beads stick to the sides or the top of the plate. In this next step, we will be moving on to the post-tagmentation cleanup. Here, we will be adding the Tagment stop buffer to neutralize the enzyme and stop the tagmentation process. Then, we will perform 2 rounds of wash steps with a Tagment wash buffer to clean up the adapter tagged DNA prior to amplification. Note that the Tagment wash buffer contains a detergent that can generate foam when pipetting. So at this step, we want to use a deliberately gentle pipetting technique with a Tagment wash buffer in order to minimize foaming. Pipetting too quickly may cause foaming and some sample loss. After the washes, we will incubate the sample on the magnetic stand to form a pellet with a magnetic bead-linked transposome. Be sure to keep the pellet in the wash buffer until the next step to avoid drying out the pellet. After cleanup, we will be moving on to the PCR amplification step. This step amplifies the tagmented DNA using a limited cycle PCR program. This step uses enhanced PCR mix, which is a master mix, along with index adapters. As we discussed earlier, the index adapter kit needs to be purchased separately, and there are several index options. During the PCR amplification step, the I5 and I7 index adapters will prime off of the partial adapter sequence that were added during tagmentation. This results in libraries with a complete final library sequence that will be compatible with all Illumina sequencers. Illumina has a list of validated thermocyclers in the reference guide. Coverage of GC-rich regions can be impacted by the model settings and performance of the thermocycler used. Something to pay attention to is that the number of PCR cycles is determined by the amount of input. The more input we use, the fewer PCR cycles are necessary. On this step, we want to ensure that all the tagment wash buffer has been removed from the samples before adding the PCR master mix. This is because tagment wash buffer can inhibit the PCR reaction and can lead to lower yield. Some best practices when handling index adapters. When we're handling index tubes, be sure to only add one index tube at a time to avoid contamination. Index kits that come with tubes include replacement caps, so be sure to replace with a fresh cap after opening each tube. To minimize contamination, this is to minimize contamination. Also, we would want to remove unused index adapter tubes from the work area. If we're working with index plates, which can be combinatorial or unique dual indexes, please be aware that each well is for single use only, even though each well includes some volume overage. This is again to avoid contamination. These plates are sealed, so we will need to puncture the foil seal before use, and we would want to store a used index plate with a new foil seal. After PCR, we are now going to clean up the amplified DNA using a double-sided bead size selection. Bead cleanup generally involves binding the DNA to the beads, washing the beads, then eluting the DNA back into solution. For the bind step, we will be adding sample purification beads to the MIDI plate. In this step, we will be using a larger volume, which is why we need a larger plate. We will either pipette to mix or use a plate shaker. Then we will incubate the plate on a magnetic stand. For the wash steps, we will bind the beads to the magnetic stand, add fresh 80% ethanol, and ensure that we do not disturb the pellet. For the elute steps, we will remove the ethanol, add the resuspension buffer, and elute the DNA from the beads. Now, let's take a closer look at this double-sided size selection in the cleanup step. The first size selection step removes large fragments. In this step, the large fragments in blue are bound to the beads and discarded. Our desired library fragments remain in the supernatant, which is then taken through the second size selection step. In this second size selection step, we remove the smaller fragments, here in orange. Our desired library fragments are bound to the beads this time. The supernatant, which contains unwanted small fragments, is discarded. The beads containing our library is then washed, and the library is eluted with RSB. This double-sided cleanup results in a library with a narrow size range, shown on the left as the red trace, This double-sided bead selection is applicable if we're working with larger inputs, including any type of large DNA, genomic DNA, cDNA, or amplicons that are greater than 500 base pairs. If we are using amplicons that are less than 500 base pairs, we have a modified cleanup protocol. In this case, we will perform just a single-sided bead cleanup with a 1.8x bead to sample ratio. This means that any fragments that are less than 100 base pairs are in the supernatant. The library remains on the beads, which we keep, and we discard the supernatant with a smaller fragment. Since the starting input is smaller in this case, this modified cleanup step ensures that we only remove the smallest fragments and retain as much of the library yield as possible. Here we will go over a few best practices for sample purification bead handling. This step is critical for ensuring that we have good library yield and the desired library size. Just like with bead-linked transposomes, the sample purification beads are magnetic and they should never be frozen. We want to bring the sample purification beads to room temperature before use for at least 30 minutes. It's also important to ensure that we're always using freshly prepared 80% ethanol for the washes, as ethanol is hygroscopic and evaporates over time. In terms of handling, we want to ensure that we vortex the beads until well resuspended, and we should use the recommended plates and magnetic sand. Also, we want to pipette up and down carefully to minimize foaming. When drying the beads, we want to allow the plate to remain on the magnetic stand to prevent potential bead loss. So at this point, we have completed all library preparation steps and we have our final library. We want to move on to sequencing as soon as possible, but first, we want to check our libraries to ensure that they look as expected before sequencing. The library quality check step is listed as optional, but we would highly recommend performing this QC step if possible. Sequencing can be costly and time-consuming, and we want to find any potential problems before sequencing. Performing this QC step is especially important if this is the first time you're preparing these libraries or if you're using a new sample type. We recommend checking the library size by running the final library on a bioanalyzer or fragment analyzer chip. Shown here are examples of what we expect the traces to look like. We expect an average library size of around 600 base pairs, which corresponds to an insert size of 450 base pairs. However, keep in mind that because shorter fragments cluster more efficiently, the observed average insert size in downstream analysis is around 350 base pairs. It's normal for the size range of the library to range from around 150 base pairs to around 1,500 base pairs. Note the slight shoulder around 1,000 to 3,000 base pairs, which is sometimes seen on the traces. The appearance of this peak can vary between DNA samples and does not affect downstream sequencing. If you're concerned about your library size or if something looks unusual, please feel free to call us in technical support or email a PDF of your traces to techsupport@illumina.com for review. The bioanalyzer or fragment analyzer lets us look at the size and shape of the library to assess quality. For quantification, if we started with 1 to 100 nanograms of DNA input, we recommend quantifying individual libraries with a fluorometric quantification method such as qubit or picogreen. In this case, we will manually normalize libraries to the same starting concentration. For 100 to 500 nanograms of input, or if we're using the blood or saliva lysis protocols, individual library quantification is not required. This is because library yield is already normalized on the beads. For optimal cluster density, if we started with 100 to 500 nanograms of input, we can pool 5 microliters of each library created in the same batch. However, there may be slight variations in final yields between different library preparation batches. So to achieve optimal cluster density, we recommend quantifying the multiplex library pool before sequencing. If we're working with blood and saliva samples, it is not required to quantify individual libraries. However, because of the more variable nature of the samples, we may still want to quantify each individual library before pooling for improved confidence. Now that we have prepared our libraries, check to see if the library sizes are as expected and normalize the library concentrations. We can move on to sequencing. Here is a table which lists the recommended library loading concentrations on different Illumina sequencing instruments. This table can be found in the reference guide. Please keep in mind that these loading concentrations are a good place to start, but some minor adjustments may need to be made based on small differences in pipetting or techniques between labs, or even differences in library sizes from what is expected. If you would like to see what an expected Illumina DNA Prep sequencing run should look like, we do have a number of public datasets available in BaseSpace Sequence Hub. After you log in, you will select the Demo Data tab as shown on the upper right side of the screen and search for Illumina DNA Prep. We have examples of runs with small genomes like bacteria or large genomes like human. We also have a range of different instrument examples. And if you have questions about these sample data sets, please don't hesitate to reach out to us in tech support. Lastly, I would like to discuss the optional blood and saliva and bacterial workflow in more detail. For the blood and saliva workflow, some additional equipment will be needed. Since these samples are biohazards, we will need a biosafety level 2 lab or cabinet with a working hood. We will also need a BSL-2 thermocycler for the lysis incubation, as well as a BSL-2 incubator that's capable of holding temperature of 50 degrees Celsius, as well as the ability to hold origin DNA collection tubes. If we're working with blood and dried blood spots, we will need to purchase the Flex Lysis reagent kit, which is sold separately. This kit is for both liquid blood and for dried blood spots, which we have a demonstrated protocol linked here. With these blood protocols, we want to use proper PPE since these samples are potential hazards. The blood needs to be collected in specific tubes, and we want to make sure that we are filling with the appropriate volume. The blood tubes should be shipped and stored between 2 to 8 degrees Celsius and processed within 3 days of sample collection. When working with this type of sample, ensure that we invert to mix the EDTA blood collection tube before adding the blood sample to the plate. The lysis protocol involves using the magnetic sand with a sample purification beads. Note that the beads and the blood have a similar color, so we will not see a clear sample as we normally would when incubating the magnetic beads on the stand. Because of this, we need to be careful when pipetting to ensure that we do not disturb the pellet when removing the supernatant. Here in the photo, you can see the white arrow pointing to the bead pellet, which has the DNA. If we pipette the magnetic beads by accident, We would just need to put the whole sample back on the magnetic stand and allow the beads to form a pellet again. There is no need to purchase additional sample purification beads for this protocol because the sample purification beads are provided in sufficient quantities with the Illumina DNA Prep kit. If using the saliva extraction protocol, we will need to specifically purchase the Origin DNA saliva collection tubes with the catalog numbers shown here on the top right of the slide. After collection, saliva is mixed with the Origin DX solution contained in the collection tube. After this, we will do a heat treatment to lyse the cells by incubating the tubes for at least 1 hour at 50 degrees Celsius using the BSL-2 incubator mentioned earlier. This protocol is expected to generate greater than 100 nanograms of DNA, which is why when we use the Salva Lysis protocol in the supplemental procedures, we expect normalized library yield. One last thing I would like to show you is the demonstrated protocol for direct bacterial colony sequencing. With this protocol, there is no need to do a liquid culture followed by DNA extraction. We can just take half of a 10 microliter loop, beat with beads before spinning down, and perform a bead cleanup. After this protocol, we will just use 10 microliters of this extracted DNA going into the Illumina DNA Prep protocol. On the bottom of this slide is a figure that shows different types of bacteria on the x-axis, and we can see the amount of DNA extracted in the 10 microliter lysate with different types of samples. With these different microbial species, we can see with a gray line that we have greater than 100 nanograms of DNA in the 10 microliter lysate going into the Illumina DNA Prep protocol. The link to the demonstrated protocol and datasheet can be found on the bottom of this slide. To finish up, I would like to share with you some additional resources for the Illumina DNA Prep workflow. The Illumina DNA Prep datasheet includes many of the figures used to explain the library preparation workflow, and it provides comparison with other Nextera kits. We also have a link to the Illumina DNA Prep support page where we can find extensive support documents, including the reference guide as well as the demonstrated protocols for dried blood spot and microbial colony extraction. Additionally, we have a link to our Illumina adapter sequences document, which has our index sequences. The index adapter pooling guide provides guidelines to help you choose color-balanced index combinations for low-plexity library pools. We also have a library preparation training video, which goes over the protocol and best practices for Illumina DNA Prep. And finally, we have a link to our base public data sets on this slide as well. At this point, I would like to thank you all for joining our webinar today. I hope this has provided useful information for you as you get started with Illumina DNA Prep.